So there are 7 chapters in this book, and these each lot just depicting the description of one chapter. Like this is chapter number 1 is sequence retrieval, and it has protein sequence analysis, from where you will retrieve the protein sequence. Usually we use UniProt for that. Then similarity search, and for similarity search we use BLAST. There are different types of BLAST software. You know that BLAST P, T BLAST X, BLAST X, T BLAST, and ABC. Then we have alignment. For alignment we usually use cluster W. Nowadays it is known as cluster omega. T Coffee is also a very good software for the alignment. Then we have sequence analysis in chapter number 2. In chapter number 2, first you know the sequence length and you have to analyze the sequence. Then you have physiochemical properties. For physiochemical properties you guys use prot param, in which you guys just analyze molecular weight, isoelectric point, a number of residues, percentage of the residues, and how many negative items, how many positive items, how many rotatable items, and many more. Then we have chapter number 3, structure analysis, in which we just perform protein function analysis by ProFunc or different types of scope, SCOP also database for this one. Active site analysis by SiteHound. Binding pocket, you can retrieve or analyze the binding pocket by different softwares. Here we have ProFunc that we used in this chapter. After that we have chapter number 4, structure prediction. You guys are very well aware of the all procedure and methodology, and I hope this is now on your fingertip. With web-based tools, you have Firex2, Ayesha, RaptorX, Swiss model, Intfold 2, and M14. Now there are so many others. And yes, I just want to mention it that this book was written in 2000, maybe 16 or 17, as I remember, and published in 2018. So we have the almost okay. So we are on chapter number 14, structure prediction. That web-based tools and homology modeling is, for modeler it's very much cited, and modeler is considered one of the best tool for homology modeling. We can also perform loop modeling by the help of modeler. There is a script available on Google. If you guys are interested, you can just download it. It's the same like you did the alignment of target and template sequence by using align2d.py script, and then you have used get model.py script for the generation of different structures. Same, there is script is available. You just have to read the script, alter it according to the nature of your experiment, mention the amino acids of loop. Suppose there are 100 amino acids in the protein sequence and you predicted the structure. 60 amino acids has very good structure, but the 40 amino acids has continuous long loop, and you guys want to improve it. So you just have to mention in the script that from amino acid number 61 to amino acid number 94, suppose it's the loop. You just mention it and remodel it. So sometimes we got alpha helices or beta pleated sheets there. So that is also an important feature of modeler. Then we have threading and ab initio. For that, threading is also known as fold recognition method. Ab initio, we start from very beginning, from scratch, and then we have to evaluate the structure. As you guys know, without evaluation you cannot say the quality, what is the quality of this structure. You have no idea. For evaluation, first of all, you guys know about your protein. Your literature should be very strong about the specific protein. You have to know where it is going to work, how it works, in where it locates. You know everything about the protein. Sometimes, or usually, people just report their observations about the protein, like it is a transmembrane protein, how many times it will cross the membrane, which amino acid will cross the membrane. And UniProt also analyze it computationally, that if it is a transmembrane protein, how many amino acids will cross the membrane and how many times. So you guys should have to study about that. There is a paper, as I remember, maybe KCNK 18. If you can check my paper, there is a paper has the protein, I remember, KCNK 18. So if you will read that paper, in that paper we have showed the transmembrane, that how many times the protein just cross the membrane. I remember the paper, yes. Otherwise I will cross check it and let you know if it is not that one. Structure evaluation is very important step. If your structure is not good, your all the experiments will be in danger. There is no reliability, so you should be very careful on this step. Our course is research methodology, so you should have to think like a researcher. You should have to think like a researcher, a reviewer. every point you should have to cross check twice. Otherwise, if you have mistake in the beginning, your whole experiment will have the wrong results. You will be in the wrong direction. That will really damage your experiments, your time, and your finance. So you should be very careful in research. And one more thing, please never ever in your whole life fabricate the data. Never ever. The results you got, please present that results. And after that, we have chapter number 6, structure visualization. There are many softwares available for structure visualization. Usually we use UCSF Chimera. VMD is also available. Pymol is available. Discovery Studio is available. MOE is available. Some are purchased ones, some are difficult one, but UCSF Chimera is a user-friendly software. And you can also analyze so many other and other analysis in UCSF Chimera, like you can measure the bond length between two items. It should be less than 3, and you can minimize the structure, or many other stuff you can do. You can also find the RMSD value of template and target structure from UCSF Chimera. I remember, and I hope you guys remember all these things. I am just recalling it for you. Then we have chapter number 7, molecular docking. There are many molecular docking: blind docking, targeted docking, flexible docking, rigid docking, and protein-protein docking, protein ligand docking. In actual, what is docking? To dock one molecule or compound with the other portion, other targeted sequence. This is known as docking. Molecular docking is at the molecular level. So protein-protein docking, in which two proteins interact with each other. As you know, protein is the basic unit. All the functions in human body is performed by proteins. The interaction of proteins with each other, interaction of protein with DNA, interaction of protein with RNA, interaction of protein with cofactors, interaction of protein with prosthetic group. So like if any the authors did not attach heme molecule to the protein, the experiment will be zero in a second. They just dock the heme molecule first, and normally, not every time, normally the compound, the novel compound usually bind near the heme, because it is considered that this where the heme is binding, it is the functional part of the protein. So more than 60% it happens. But we also have other examples. Like I was 2 or 3 years back, 2 years back, I was working on a project in which we also have this type of prosthetic group and the cofactor that helps the protein to be functional. And as we assume it that it is the functional part of protein, so the external compound will bind there, but external compound did not bind there, bind somewhere else in the protein and disturb the interaction. So you should also keep in mind about all these scenarios. You are a researcher, you should have to think like a researcher, you should have to focus on all the points.